Oh, it's still broken. That drives me nuts. All right, welcome back. Let's talk a little here about neurogenesis. All right. This should be a short one. I know we've had some long ones this week, but it won't be near as long as the other ones. All right, so this is a lot. So bear with me as we try to a lot of these. Ever since we got into like the last couple chapters, like this one thing that bugs me with the textbook. Like I know it's a complex process, but man, some of these figures, there's a lot of information on. So we're going to do our best to as clearly as possible show what's going on here. First thing I want to point out here is at the bottom is the time. So we have the left-hand side is how everything is pre-neurogenesis. As we move to the right, we have neurogenesis, and then we have late neurogenesis and gliogenesis. Okay, so this is as time progresses. Okay, so as neurogenesis, or as the neural tube matures, the product of the neural epithelial cells, that's NEC, neural epithelial cells, become radial glial cells. All right, so RG, radial glial cells. Neural stem cell, or these are neural stem cells that undergo symmetrical and asymmetrical division, right? So NEC cells can become more NEC cells, or NEC cells can become these ventricular radioglial cells, okay? So divisions of these cells occur in the ventricular zone. So on this axis here, as time progresses, we get these zones forming. Remember, we had all these different zones that we talked about before in previous lectures. So everything under this line is the ventricular zone, okay? So this happens in the ventricular zone early in pre-neurogenesis. Okay. As the progenitors delaminate from the ventricular zone, so they are moving, just like we talked about with neural crest moving, these cells also move. These two layers form the germinal strata that generate the neurons that migrate to the cortical plate and form the neocortex, so that they start forming these cells here during neurogenesis. So we've got these ventricular radioglial cells here. Now those ventricular radioglial cells will reproduce and form another radioglial cell and a neuron. These purple cells, right? These are deep layer neurons. Those neurons will become, up here, cells in the cortex, right? Okay. So as you can see later on in the intermediate stage of neurogenesis, those ventricular radioglial cells, those will form intermediate progenitor cells, right? And those can stay in the ventricular zone or into the outer subventricular zone. They'll migrate. So those are the orange ones here. Those orange ones can also differentiate into neurons. So we had these ventricular radioglial cells that can turn into neurons, or an intermediate progenitor cell. They don't asymmetrically, so this is asymmetrical, right? Because it's making two different cells. They symmetrically only make more neurons, right? They don't make more IP cells. So they'll make more neurons in the during the intermediate progenitor, during later neurogenesis. Then we have these outer radioglial cells, which is even later in neurogenesis, and that's where these radioglial cells can form the outer radioglial cells and one intermediate progenitor. The outer radioglial cells can make another asymmetrically another outer radioglial and one intermediate progenitor, etc. And so as you can see, as time progresses, we get more and more layers where during neurogenesis we start to form the outer subventricular zone, the intermediate zone, and then we have the cortical plate neocortex, where we have deep layers and upper layers of that neocortex, etc. So as time progresses, we have increased differentiation and migration of these cells. Okay, so we also have this neuron-glia interaction that occurs in the mouse. And so we talk about this migration. So these neurons migrate on these glial processes. So for them to move up layers, they like kind of wrap, you can see them on here, kind of wrap and climb along this leading process of the neuron, climb along this glial cell. And it can go pretty quick. So it can go up to 40 millimeters, which isn't like big when you think of it in terms of us, but for a neuron, 40 millimeters in an hour, it can climb across these glia. So this is just kind of an experiment that was done to determine, this was in a ferret cerebrum, how these cells migrate. So this was embryonic day 29. They used this tritium labeled neurons. So tritium is, so it's like a radionucleotide that they put in there so they could map where these neurons are going. So at day 29, they labeled everything in cortical layer 6. So you see a high percent of cells that have this radio label on it. And then they came and checked back. So postnatal day 1, so after birth, they found that all those cells that were in layer 6 at that time point, at day 29, had migrate to levels 2 and 3. So this is just kind of a proof of concept that we do know experimentally that these neurons start in one layer, and then at birth, or a day after birth in this case, they're able to move and migrate. So this is, again, this kind of an image of what was happening in that last ferret experiment. So this is delamination of the cortical laminar identity in the ferret cerebrum. When early neuronal processes, which are the dark blue here, are transplanted into the older ventricular zones, they migrate to layer 6. So what they did is they put radio-labeled nucleotides, or grew these neuron cells in these radio-labeled nucleotides. So they didn't do this in situ. They could do this in petri dish. So they implanted cells, neurons that were grown with this radio-labeled media, tritium-labeled media, into layer 6, and then found that after birth they were in layer 2. So in this, they're showing that they grew these cells, implanted them in layer 6, and then found that they had migrate to layer 2 after birth. That's just an added picture of what's going on here. Why do I have two of the same slide? What's going on here? What's this slide mean? Okay. We'll skip that slide. It doesn't give us much more information. All right. So this is just kind of showing this asymmetrical division of radioglia. So remember, we had those radioglia that can form more radioglia, or they can form a different cell type. So this is regulated by this gene called PAR3 and also Notch. So if there's, depending on if there's high Notch or low Notch, changes the cell division. So PAR3 as well. So PAR3 protein function in a radioglial cell let's see, helps with this adhering there to the lumen tube. You can see it's expressed right here on the very distal end of this neuron. The dynamic distribution of PAR3 in these luminal stem cells regulates the synthesis of Notch. So it's what determines if there's high Notch or low Notch. So at mitosis, PAR3 becomes localized primarily at one end of the two daughter cells. So at mitosis, oops, sorry, that's what I just read. That daughter cell will express high levels of Notch and remain, or that daughter cell that does express high levels of Notch will remain a stem cell, and the cell receiving less PAR3 will express less Notch and become a neuroprogenitor cell. So in essence, what is happening? So PAR3 is expressed at this distal end. So we'll go through S, G2, metaphase, and then anaphase, telophase. So it's starting to split here, right? So we've done, we've pulled apart. You can see right here, we're pulling apart the spindle fibers, are pulling apart the chromosomes here, and they're going to be segregated into two different nuclei, and we're going to split the cell in half, right? So if one of the cells, because that PAR3 is located on a certain part of this membrane, in this case it's shown to be a lot bigger of a region for some reason, but if one of the daughter cells inherits a high amount of PAR3, that will mean, remember PAR3 regulates Notch, that means that it's going to have a high amount of Notch, and so that will form it into one cell type. If one of the daughter cells did not get a lot of PAR3, in this case it got very little and it just has this N-cadherin around it, that means it'll have a low amount of Notch. And so that will make it into like one of these glial cells or Schwann cells or something, like whatever, asymmetrically divided. So 1,000-yard view for this. Like I said, some of these are complex. PAR3 is expressed in a small part of the cell wall or the cell membrane. When this asymmetrical division happens, the cell that receives or maintains or keeps that part of the membrane that has a lot of PAR3 will have high Notch and become one type of cell. The other daughter cell that did not split off the membrane with that PAR3 will have low amounts of Notch and will become a different cell type. Okay. So lastly, this is showing how this works with microscopically, I guess is the best way to put it. So the very bright green down here is PAR3. And then red is the nuclei, and then the rest of the green, I don't know, they didn't say what the lower green. The bright green is PAR3. Doesn't say what the rest of it is. Just cytoplasm, something in the cytoplasm. It could be off target. It could be other PAR3. I wouldn't expect it. Just like non-concentrated, but it could just be off target labeling too. Anyways, so if you look at this, we have the cell, it's undergoing cell division, and you can see that this daughter cell on the left got a whole bunch of bright green. And this cell right here, which is going to become this cell, you can see like in between these arrows is where that cell membrane is going to split, does not have that bright green, right? So here we have this segmentation of PAR3, a ton of it down here. This cell does not have near as much. So that means that these two cells can then differentiate asymmetrically into two different types of cells, whether one's a progenitor cell, one becomes a neuron, or one becomes an intermediate progenitor and one becomes a neuron, whatever from that first image, however asymmetrically you want to divide this. This is like an example of that. Okay, that's it. Short one, 15 minutes, 10 minutes shorter than the last few. That is it for neurogenesis. Thank you for watching. I hope you enjoyed the video, and I will catch you in the next one.